Question number 121. An individual affected with Turner's syndrome. First option, lacks X chromosomes. Second, has an additional copy of X chromosome. Third, is monosomic due to presence of only one X chromosome. Fourth, has trisomic condition of an autosome. Individuals affected with the Turner's syndrome are sterile females with chromosome constitution 44 plus XO. So, these females are monosomic because human females have chromosome constitution 44 plus XX if they are normal. But this X is not present. There is a loss of this X which leads to a condition of Turner's syndrome. So, these females lacks X chromosomes? No. They have one of the X chromosomes. Has an additional copy of X chromosome? No. It has one less X chromosome. Fourth, has trisomic condition. Trisomy means when there is additional chromosome. Means in human females, if there are 46 chromosomes, in trisomy it will be 47. But here, the total number of chromosomes are 45, not 47. So this is not the trisomic condition. Third option is monosomic due to presence of only one X chromosome. This is correct. So the correct option for the given question is option number 3. Let's move to the next question. Question number 122. By performing a series of experiments that showed the effect of R and S strains of Streptococcus pneumoniae on mice, Griffith concluded that first option, protein digesting enzyme does not affect transformation. Second, DNA is more stable genetic material than RNA. Third, DNA replication is semi-conservative. Fourth, non-virulent bacteria were transformed by heat-killed virulent bacteria. Griffith conducted experiment on R and S strains of Streptococcus pneumoniae, and it was the transforming principle. So, see the statements now. Protein digesting enzyme does not affect transformation. It is true statement that transformation is not inhibited by the protein digesting enzyme. It is just inhibited by the DNase, which is the DNA digesting enzyme. But this was not the conclusion of Griffith's experiment. So this option is ruled out. Second, DNA is more stable genetic material than RNA. It is also true, but not the conclusion of Griffith's experiment. So this option is also ruled out. Third, DNA replication is semi-conservative. It is also true that DNA replication is semi-conservative. Many scientists prove that also. But this is not the conclusion of Griffith's experiment. So this option is also ruled out. Now, fourth is a non-virulent bacteria were transformed by heat-killed virulent bacteria. Non-virulent bacteria were R-strain bacteria, which are rough and they cannot cause any disease. Heat-killed virulent bacteria are the S-strain bacteria and they can cause the disease. So, the R-strain bacteria were transformed by S-strain bacteria which were heat-killed. This was the conclusion of the Griffith experiment. So the correct option for the given question is option number 4. Let's move on to the next question. Question number 123. Deoxyribonucleoside triphosphates serve dual purposes in DNA replication. In addition to acting as substrate, the other function is similar to first option, DNA polymerase. Second, rRNA. Third, topoisomerase. Fourth, ATP. DNTPs, deoxyribonucleoside triphosphates, also called DNTPs, they serve dual purposes during DNA replication. First, they are substrates, and second, they provide energy for DNA replication. So they act as ATP also. So the correct option for the given question is option number 4. But what about the other options? DNA polymerase, it's an enzyme which helps in DNA polymerization. Topoisomerase, again, it's an enzyme which helps in removing the strain in the DNA string during the DNA replication. rRNA is a component of ribosome. It's ribosomal RNA. Let's move to the next question. Question number 124. In lac operon, First option, promoter gene interacts with regulator molecule. Second, regulator gene possesses the site for RNA polymerase attachment. Third, LAC-Z codes for an enzyme that increases permeability of cell to beta-galactosides. Fourth, the I gene codes for a protein known as repressor protein. What happens in lac operon? Lac operon is present. Lac operon takes place in bacteria for the metabolism of lactose. First option, promoter gene interacts with the regulator molecule. No. Promoter is the site for RNA polymerase attachment. This site does not interact with the regulator molecule. Second, regulator gene possesses the site for RNA polymerase attachment. No, the site for RNA polymerase attachment is promoter. Third, LAC-Z codes for an enzyme that increases permeability of the cell to beta-galactosides. What is a beta-galactoside? Lactose is a beta-galactoside. It's a sugar. And LAC-Z gene codes for an enzyme which increases the permeability of the cell to the lactose. No, this is not correct. In lac operon, there are three structural genes, Z, Y, and A. The Z codes for beta-galactosidase. It's an enzyme which can break down lactose into two sugars, which are glucose and galactose. This Y codes for permease, which increases the permeability of the cell for lactose. So this statement is incorrect. Fourth, the I gene codes for a protein known as repressor protein. I gene is the constitutive gene of the lac operon and it codes for a protein known as repressor protein. It is correct. This repressor proteins binds to the operator. So the correct option for the given question is option number 4. Let's move to the next question. Question number 125. For which of the following qualities Saccharum barberi was selected to get desirable qualities in new varieties of sugarcane? First option, ability to grow well in low winter temperature. Second, high sugar content. Third, thicker stem and reduced leaves. Fourth, soft and branched stem. Saccharum barberi was originally grown in North India. So it has ability to grow well in low winter temperature of North India. But it does not has high sugar content. It has a low sugar content. So this option is ruled out. It has thin stem, not the thicker stem. So this option is also ruled out. It does not have soft and branched stem. Hence this option is also ruled out. So just to get the desirable qualities in new varieties of sugarcane, Saccharum barberi was selected just because of its ability to grow well in low winter temperature of North India. So the correct option for the given question is option number 1. Let's move to the next question.